So it's a great delight to welcome you back to the reading group, Jonathan. And I understand that Joel will also be joining us here in a little bit. We're discussing a paper of yours that I guess it's still at the preprint stage. Is that right? Yep.
Yeah, that's right.
So we're looking at some really detailed analysis of the early samples out of Wuhan. You go into lineage A and lineage B, and I thought I'd just kind of start with a real softball question, and if you could just tell us what lineage A and B are. As best as I can tell, it's just a couple of nucleotides different. Is that right? And so one of the key conclusions that you're suggesting here is that there's more than one event. It's not just kind of a single clade flowing from animals to people, but you've got evidence of at least two events. Am I reading the basics right here?
You are, yeah. So lineage A and B are kind of the first two well-defined lineages of the pandemic. And we saw evidence of lineage B first, and it really took off early on. And most subsequent lineages that we've seen since the start of the pandemic fall into lineage B. Lineage A arose just a little bit after lineage B, just a few weeks after. But it's actually a little bit more similar to closely related bat viruses. It's actually only two mutations similar at sites 8782 and 28144. But it didn't take off the same way lineage B did at first. And it was kind of that discrepancy that really piqued our interest and made us want to look more deeply into that. And yeah, by the end of the paper, we kind of conclude that they were likely separate introductions from an intermediate host.
And maybe just another like super basic question. How do we know that this isn't just sequencing error or something like that? How do we know that there's actually a different lineage?
Sure. So I mean, if it were just like one or two genomes, that would be one thing. But even in our data set, it's like a third of it, and we can examine the integrity of these genomes pretty well. And when you do so, you see that they are being correctly identified at those two specific positions. Last time I was here, we talked about one of my other preprints that looked into what we called intermediate genomes that matched one at either lineage B or lineage A at one site, and then the other one at the other site, making it look like one of the lineages was evolving into the other one. And we were looking at those to see perhaps maybe those weren't real, or maybe there was some sort of bioinformatics or sequencing artifact or something there. And we eventually saw that there is evidence for that across the early intermediate genomes early intermediate genomes. And then that's kind of really how this paper got started, right? Of course, we're not trying to correct for every single genome. We're trying to correct for genomes that we are specifically interested in. Joel has just joined, so hello, Joel.
Welcome, Joel. So we already got started a little bit, just asking super basic questions. And maybe one other really basic question. So the paper is focusing on what I understand to be 15 non-recombinant sections of the genome rather than the full genome. So first I was just wondering, why go for the non-recombinant sections? Is it possible to, tease out the different what's going on with the recombination dynamics? But yeah, I guess why not look at the whole genome? Why look at just those non-recombinant sections?
So we do look at the whole genome. I wouldn't say the paper's focused on the 15 non-recombinant regions. We're focused on the whole genome. We use the 15 non-recombinant regions for a specific technique to help root the phylogeny. Normally people do something that's called outgroup rooting, where you take a similarly related virus and then you use that to orient the topology of the tree. Alternatively, what you can do is you can try to understand what the ancestor might have been. So the related virus is a taxa, right? It sits on the outside somewhere in the tree. But we might be actually interested in that internal node, the most recent common ancestor of both everything that we have and that other virus. And so what we tried to do was construct that internal node, that ancestor of SARS-CoV-2. But coronaviruses are very recombinant. And so we took 15 non-recombinant regions, which is the whole genome. It's just those are the 15 regions that are inferred to not be combining with each other based on the genomes that we have. There could be more recombination, there could be less. It's like, it's hard to be perfect about it. But once we have those 15, we then basically infer that most recent common ancestor, that ancestor of SARS-CoV-2 and the closely related bat virus for each of those 15 regions, because it's not the same for all 15. And then once we have that, we can string it together. We now basically have what we call a recombinant common ancestor of SARS-CoV-2. And then we can use that to orient the topology of the tree instead of a related virus. That's really kind of the point of it. We look at the whole genome for both, the bat virus as well as all the human viruses. It's just a specific technique that we use.
Thanks. And yeah, definitely want to open it up. There's lots of people better qualified than me to be asking the questions today. So, if you have a question, feel free to just unmute yourself, type it in the chat, or raise your hand. But I do have more questions. They might not be as well informed as some of the other people on here. And I'm just bringing up the paper too to try to get it in another window. And maybe it's really, so I don't have any background in bioinformatics, so like some of this stuff I just simply can't follow. But sticking with that idea of trying to root these phylogenies, I was struck by it seems like a single most recent common ancestor that you're going for rather than multiple ancestors, and I'm just wondering, just in the context of the frequent recombination, if it's maybe should we be looking for multiple common ancestors rather than just one?
Sure. So I guess moving forward, let's break this up into two terms, just for everyone's ease. When we refer to most recent common ancestor, we'll just reserve that for SARS-CoV-2, and for that ancestor of the related of a bat virus or some other closely related virus in SARS-CoV-2, we'll call that a distal ancestor. And so when we're talking about the distal ancestor, right, of SARS-CoV-2 and then a closely related bat virus, with the non-recombination, there's 15 of them, right? And so we infer those 15 genomes of that distal ancestor, which I just described. For the most recent common ancestor of SARS-CoV-2, yes, there's recombination of SARS-CoV-2 as well. We've seen that in the past 2 years. But we don't really have evidence of that in the first 2 months of the pandemic. That said, we do still infer 2 most recent common ancestors for SARS-CoV-2 rather than 1, because the paper is really going to show that there were likely at least 2 introductions, and the 2 most recent common ancestors would be of lineage A and B. The most recent common ancestor of A and B together, is also part of the paper, and can match the most recent common ancestor of just one of the lineages. But there's just like a little bit of nuance there.
Mm-hmm. Okay, it's hard to call on Jonathan, but Jonathan, one of the other people named John or Jonathan in the room.
I'll ask for forgiveness for also possibly asking a very ignorant question, but I am curious if, I guess, as I understand it, you're looking at genomes that are existing more or less at present and kind of calculating the possible tree that they belong to. And I'm curious if that those calculations involve kind of standard algorithms that everyone uses when mapping viral trees, or if you all are kind of doing the kind of calculating the writing an algorithm yourself that kind of if there's something unique to the situation. And I'm also curious if when we're creating viral trees as opposed to trees of life of cells and multicellular organisms, if the algorithms are the same, or if there are specifics to the situation that require using different kinds of programs.
Sure. So I'll answer the first part, and then I'll let Joel go for the second part. But with our paper, most of the algorithms are pretty well established already. And all the ideas, I would say, are already pretty well established as well. The new algorithmic developments in our paper would be first, I mentioned how we used this recombinant common ancestor to our topology, or to shape our topology a little bit. But normally people do outgroup rooting, as I mentioned, with that closely related virus. And so there hasn't really been a clear method on how to use essentially this ancestor node or this ancestor genome to root to root a topology. And so we worked with another lab to help expand the phylogenetic tools available to allow us to do that. And so that was one of the developments. And then the other development is that most phylogenetic inference models are what we call reversible, meaning the substitution model is reversible, meaning that when you have a mutation from like A to T, a lot of these models assume that how often you go from A to T is similar to how often you go from T to A. But that isn't necessarily true. And it's certainly not true with coronaviruses in humans, where there is a mutational bias of C to T. And so we had to make sure that we had a substitution model that was accounting for the C to T bias, likely due to some antiviral host properties. But that was an important thing that we needed to capture in our model to make sure that we could properly infer the most recent common ancestors of our topology, because some of the most important mutations early on were those C to T and T to C mutations. And if you don't have the proper rates for each of those, you might incorrectly assign rates to both of them, or at least to one of them, and then incorrectly infer what the shape of the tree and what that ancestral genome looks like, because you have an incorrect substitution model with your phylogenetic inference. And I would say, so those are really the two techniques that we had to develop for our work. And then for tree of life stuff, I'll give it to Joel.
Yeah, hi, Joel Wertheim. Apologies for my lateness today. So there was one other, I'd say, methodological insight that actually came from Jonathan's previous paper, looking at the timing, which was how to do sort of TMRCA inference before the TMRCA, because the TMRCA represents the ancestor of all sampled genomes. But of course, you could have sort of a stuttering transmission, like your first person infects two people, only one of them goes on to infect everyone else, and you have one dead end chain. Now, that may just be a day or a week, but the question is, how long could that have been? So what we did is
TMRCA is time to the most recent common
Oh,
ancestor.
sorry, I didn't. Yeah. So you're basically the time. What happened before the genetic ancestor, and using what are basically standard coalescent approaches, basically understanding how long does it take to trace back to a common ancestor. You can say how long could you have gone forward in time before you get to the point in time where you would on average trace back to the common ancestor. And coalescent theory is sort of very well established, mainly used for population genetics, but also part of the software that Jonathan's been using. But the sort of beast phylogenetic software typically ends that coalescent process at the genetic ancestor, at the TMRCA. So we sort of look to see how far could you go before you got to that point, and then that gives us sort of that lead time before the genetic ancestor, what we call the primary case, which will very often predate that time of most recent common ancestor. So that was I think, an important methodological sort of innovation that allowed us to estimate something that people always assumed existed, but no one had really tried to quantify before.
I also pause here very briefly because so in our the last time that Joel and I were on, we called it the index case. And the index case is the first person we know who was infected with the given virus, in this case SARS-CoV-2. And we were actually previously referring to the primary case, even though we called it the index case. The primary case is the first person who was actually infected by the given virus that then led on to the chain of transmission. We might never know who this person is. And so just, yeah, if you watch our last video and this video, we meant to say primary case last time, and that's what we mean by primary case now as well.
And then to answer your other question about are viruses different than the rest of life? Sometimes. So as a part of all of this, I recently reconnected with some old phylogenetics and math colleagues of mine who basically helped characterize this unique sort of phylogenetic space that viruses like SARS-CoV-2 or Ebola or say an HIV outbreak sort of inhabit, where because if your sampling is so dense over a very short period of time, you basically capture every mutation along the way. And that puts us in what people before me have termed a near perfect phylogenetic space, where you can essentially infer the phylogeny without ambiguity. Now, Jonathan, when I came on earlier, he was talking about all these errors in early genomes, and clearly sequencing error can lead to sort of confusion in the inference, but he actually did a great job cleaning that up. And what it shows is that this tree is actually very understandable, and you can really track, as we've seen with the rise of variants, every mutation along the way, which allows us to do things and allows us to be a lot more confident in the tree we're inferring. It allowed us to infer this ancestral virus, the most recent common ancestor sort of genomic haplotype, with far more precision than even the developers were expecting. Once we had the results of that, he threw up his arms. He says, well, I can throw out the second half of what I was planning to do because this is so much easier. There's one genome or three genomes, and there's no sort of weird combinations going on. So that allows, and then also for those of you who sort of build trees in your spare time, it means that you don't want to go around bootstrapping these trees because you have one change that supports an entire clade, and the odds of picking that one mutation when you go and you do your sort of phylogenetic bootstrapping is 1 out of 63 out of 100 times, which means your bootstrap support for your one mutation defined clade is 63. So we're very different than the rest of true life, or very, very distantly related viruses, and that's, I think, a sort of novel, sort of very niche methodological insight that came out of looking at SARS-CoV-2 trees. But since I'm here and you asked, I thought I'd share.
Maybe just sticking or going back to kind of some of the small details about lineage A, and also some of the assumptions you're making about kind of the direction of these changes. Could you walk us through the evidence that this is more closely related to a bat? Is it just, I mean, why not just say like, okay, you've got this incredibly intensively studied set of genomes, you've got these two mutations, like, how do we know that, you know, what you're calling here lineage B didn't come first, and then there's just like those 2-point mutations that then kind of created this other population?
Yeah, I mean, really, it's just that, like, that would be very unlikely. You have your given ancestor, right? And so we can, as we talked about with that distal ancestor, we can infer that it's also has those same 2 mutations or nucleotides that match lineage A. And so then if you were to have lineage B come first, right, and then it evolve into lineage A, that means you just have to have two mutations from the distal ancestor to SARS-CoV-2 to get to lineage B, and then another 2 mutations going back to get it to lineage A. And so that's 4 mutations in a given time span, and especially 2 mutations basically in the span of like a few weeks, which, you know, 1 over 30,000 times 1 over 30,000, approximately, for those 2 specific sites is just extremely unlikely. And so it's not impossible, right? Like, and some of the models do infer that as what happens. But when you also combine it with the epidemiology, where you do actually see what is more distantly related coming first, the more congruent hypothesis just is that it's not what happened, that you didn't have the evolution of one toward the other. But Joel might want to add some things.
Yeah, so that, I mean, just great explanation. I'll add some details around the fringe there. So A and B, so there are only 2 mutations different, and 2 mutations sure doesn't seem like a lot. But if you just look at the molecular clock, you know, the changes that accumulate through time, which for SARS-CoV-2 has really been remarkably consistent over the entire pandemic, with the exception of the branches that led to Alpha and Omicron. Everything else is basically.
Oh, you want to explain molecular clock?
Oh, yeah, sorry, I didn't. Oh, yeah. So molecular clock is basically the number of mutations that accumulate over time in this sort of, well, clock-like fashion. And SARS-CoV-2 gets about 2 mutations added to the genome along every single lineage per month, about 24 per year. And that's just a function of the errors that the virus makes while replicating and occasionally adaptation. Now, if you look to see sort of, and the molecular clock allows us to trace back to that time of most recent common ancestor. So what you end up with is a clock that tells you very, very strongly that the ancestor of all of SARS-CoV-2 is B, and that lineage A actually is not the ancestor according to the molecular clock. It's sort of buried in the original preprint, but it's fairly emphatic that you cannot root SARS-CoV-2 on A and have a sort of consistent molecular clock, which is unexpected because you said it's 2 mutations closer. So one option is that, yeah, B just evolves into A. But when then we go and we root on this sort of recombinant ancestor or outgroup, it then very strongly pulls it towards A. So basically A is out of time. And that's, I think, something that not only people have said, well, yeah, B was there first, and A, wasn't seen around the market. But the question is, it's really more emphatic than that, is that A is a viral lineage that is out of time compared with B. And the way to explain that is to say, well, A and B aren't on the same clock. They basically start on different starting points. And that if B gets to start going first with its 2 B mutations, and then A gets to start going a week or 2 weeks later, then the molecular clock sort of lines up for the both of them. So it's not just that it's 2 mutations, but it's them when they were sampled and relative to each other to basically make the clocks for both lineages tick in sync.
Thanks.
We have a question.
Yeah, go ahead, Frederic.
Yes, thanks for making bioinformatics accessible. It's a very fascinating domain. It seems that your article gives a strong argument against lab leak hypothesis, because one of the arguments of those who supported this hypothesis was that there was a single spillover event, which could suggest that it comes from a lab. But so do you think that if there are several spillover events, it tends to discard this hypothesis?
I mean, my opinion or what the response will be? Yes, my opinion is that none of this is consistent with a lab leak. Multiple introductions of two non-identical viruses around the market, sort of radiating out, are entirely consistent with
happening first.
Yeah, entirely consistent with it with a natural spillover. I mean, basically, our timeline looks like, B jumps in at the market in late November, A jumps into the market slightly later, mid to late November, then late November, A jumps in. December 23rd, all of the farms upstream in Enchi close. Between December 23rd and January 1st, all of the animals, like common raccoon dogs that we know were at the market in November and October of 2019 are no longer at the market. Then the market closes. And I mean, I think you have a very natural progression of, oh no, we have another SARS in our hands. Quick, get it, let's stop this and get rid of all the animals. Unlike last time where they kept selling the animals for a year and a half. Let's end that, and then the spillover stop. Now, that said, there's nothing to preclude them from saying, well, yeah, but they had two different strains of the virus in the lab and they brought them to the market. And and we finally found a sample with SARS-CoV-2 RNA and raccoon dog or mink DNA in the same environmental sample that the Chinese CDC has. Someone will say, yeah, but a lab worker went, they shopped at the market and we had reverse zoonosis. We infected the animal, and there's no end to this. So although in my mind, these two preprints are sort of, nail in the coffin of lab leak. Not that it was sort of tenable before, but I think we can move past it. I have no illusions that this will end the sort of debate, or really it may end the debate, but it will not end the conspiracy, because there are endless permutations, reverse zoonosis, multiple jumps, multiple lab workers infected. So I have no illusions of Twitter clearing itself up anytime soon.
Do you have a follow-up, Frederic?
And it's not possible to assume that these two events could come from different strains coming from the lab, because that's very unlikely that two lab workers would come to the same market with two different strains.
I mean, we're talking about a tiny little market in the middle of a city of 11 million people. I think, of all the gin joints in all the world, they wander into that one. I mean, look, is it possible? Yes. Is it? I mean, if I tell you that, we have a novel coronavirus in a place that, with poor sanitary conditions that sells host animals for it, and no evidence that, this thing existed anywhere, and no close relative in a lab, and frankly, bat viruses, although in the aggregate are distantly related, piece of them are pretty close to SARS-CoV-2. So, it's not hard to imagine sort of a bad ancestor existing at some point, in Yunnan or Laos and making its way, just like SARS-1 did into Hubei province, because people tend to forget that SARS-1 infected civets in Hubei, although no humans there, definitely civets. So, I mean, like I said, you can never disprove it, and it endless permutations. But what's the most likely origin of a zoonotic virus? Zoonosis. And now we have to account for two of them equally radiating out from the market in succession.
Oh, I think you're muted, Frederic. There's another question in the stack, and I've also got another question. But if you have a quick follow-up, go ahead.
Yeah, very quickly. do you think, since you seem to assume that raccoon dogs would be the reservoir, the intermediary animal, do you think it's possible to experimentally confirm your hypothesis by showing how raccoon dogs could produce different strains of SARS-CoV-2?
I mean, they've been experimentally infected with them and are capable of transmission. Of course, I don't know if it's raccoon dog. I mean, they had lots of animals there that could host this virus. They had mink, they had deer, and of course we know that from SARS-1 that raccoon dogs and civets and mink are all sort of capable of having a virus and allowing it to mutate. Now A and B, we can call them different strains, but really they're just two mutations that don't really seem to have much fitness consequence at all. So I think they're just, we can sort of see them as neutral markers in this case, in which case you just have, a shallow reservoir of viral diversity in some unknown animal or species or multiple species at the market.
So maybe before I pick up on the other Jonathan's question here, I just wanted to ask a long question that kind of focuses on first some of the data in table S7. So that's the one where you're estimating the number of infections of lineage A and lineage B. You're looking at numbers, and this is for people who are looking at the paper, page 55. And you're looking at December 2019, the 15th of December, estimating, in my mind, what are pretty low numbers of people infected. So, part of the question is just like, we know that this virus doesn't produce serious, medical problems, and most people aren't hospitalized who get this even without a vaccine. But I'm also kind of asking this question with, the work of Len-Fa Wang and Suprapon and Thailand, like who've been studying, the circulation of SARS coronaviruses for decades now in Southeast Asia. And just wondering, are we? I guess I'm not yet convinced that both of the Wuhan stories are, I think there was a lot of tourism to Southeast Asia from Wuhan, and the possibility that the market was a super spreader event rather than the place where it originated is, at least in my mind, and some of the virologists in Southeast Asia's mind, like a probable thing. So just looking at that that figure on page 55, could you walk us through how you got to those numbers? And to me, just the basic math, again, this isn't my field, but knowing that I think Sasha would be a good source on this too, but I think it's from 1 to 3% of people with SARS-CoV-2 infections end up needing serious medical attention and hospitalization. So last week we didn't record this for YouTube, but Dominic Dwyer, who is part of the WHO team, spoke with us, and one of the things that he's been talking with me about and with the group about is how in some ways the fever hospitals of China are very good, like exceptional in some ways, at least compared to what's in place in Europe or the US. But this particular facility in Wuhan only had the capacity to engage with dozens of patients, maybe 100 at the most, which is tiny in a city that's larger than New York City. So his suspicion is that there was a lot of cryptic transmission before it was detected. So again, just I'm asking about that table in particular, and the speculation that there was cryptic transmission a long time before it was picked up.
Yeah, so I think it might be useful to start with the models that we use and then work our way backwards from there. So we talked about inferring the time of the most recent common ancestor. And that's just one of the models we use. The rest of it is simulation based. And so we use simulations to figure out how many cases there are at different times. Our approach fuses the timing of the most recent common ancestor with the analogous time in our simulations to figure out how many cases there would be by the time of the most recent common ancestor, as well as past that. And the case counts that we get, or rather the infection counts that we get in that table, they're very low because we are basing, once we link up the simulations with the time of the most recent common ancestor, we are basically able to see that there were just very few cases by that time period. And that makes sense given that our model times the introduction events to be roughly in mid to late November. So if you have a start in mid to late November, it is of no surprise that then you have very low infection counts in December. Exponential growth, it grows very quickly after that, of course. But I would say the numbers are very consistent with the models that we use, but I would also say that they're very consistent with what we actually see in December in Wuhan, where, right, by January 2nd, I think we have something like 41 or 42 recorded hospitalizations. Our model captures that. So we're able to actually reflect known numbers in Wuhan in our model. So that gives us some extra confidence of what we're seeing. We assume that certain numbers of people are actually caught or identified by public health authorities in our model, that certain numbers of people are hospitalized with severe illness and so on. And those rough estimates also match what we've seen in the literature. And so the way we've calibrated our model allows it to recapitulate what is known about the pandemic at the start. Going on to like cryptic circulation though, or cryptic spread, our model basically showcases that by the time of the most recent common ancestor, which on average, like in the phylogenetic inference, is usually around December 9th, there are at most like a few dozen infections. Even at the most extreme scenario, it's usually not much more than that. And we know when the time of the most recent common ancestor is. That has nothing to do at this point with case counts. That has to do with the genetic diversity. And so we can inform our understanding of cryptic spread now, not just with case counts, but also with the genomic diversity and the phylogenetic inference. And I think that is a pretty important part of the paper that we try to expand on a little bit in our revision, that you really aren't likely to see huge numbers of cases or infections by the time the most recent common ancestor. And since we know when the time the most recent common ancestor is, it's kind of hard to imagine that there was long cryptic circulation before then, even in the most extreme events where you have the slowly petering virus before it really takes off.
Yep.
Super spreading, I'll let Joel go. Yeah.
I was going to say another important point there is extinction. So most viruses that we introduce into the model go extinct 78% of the time. If you look
67% in the preprint.
67 in the preprint. Spoiler alert. But it was 70% in our previous paper, and then if you look at the excellent studies looking at the introduction of the virus into the UK, either at the beginning of the pandemic or the Delta wave, half of those introductions go extinct or more, without giving rise to any cluster. They just go extinct. So sort of part of that, fat tail distribution, that tendency for super spreading means, yes, some people infect a lot of people, and that's in our model, but most people infect no one. So you can't have long-term low-level transmission without risking the virus going extinct. Either you have to get big and establish an epidemic, or you can be low and then take off, but you can't sort of be low and somehow infect a lot of people and not be noticed. Because as Jonathan said, the genetic ancestor is sort of independent of all of these observations. It's something that we can infer retrospectively, like like we've done for the 1918 flu. Like you don't have to be there to capture that genetic ancestor. Just a few samples should do it, or at least 20. So what we see is that this genetic ancestor by, end of November or December, it exists. Now you can't have widespread cryptic transmission and in say October or November and a genetic ancestor that exists later. You have to get one or the other. One leads to the other. Sorry to misspoke. So it's inconsistent with sort of the epidemic modeling to miss this genetic ancestor. So I would say we can say what if, but there's no sort of transmission dynamic that allows you to not go extinct, exist in widespread, sickening a lot of people in October and November, and then not have a genetic ancestor until December. It's inconsistent sort of with our understanding of sort of transmission dynamics and evolutionary theory. That said, they went back to for thousands of people were hospitalized with influenza-like illness in Wuhan prior to the pandemic, and not a single one of those samples was positive. The WHO went back and tested, and there's no evidence of SARS-CoV-2 there. The only evidence we see earlier in the world is in wastewater in Spain and Brazil, and sort of these results from hemi-nested PCR in Italy, which all seem like false positives to me. So I don't think there was cryptic spread. There were clearly people infected, but there wasn't widespread illness because you just couldn't infect enough people to do that.
On the serology from Wuhan, actually, people like Dominic Dwyer would disagree that there was adequate serology done prior to the pandemic. And one of the points that he made last week was that the samples were destroyed after they were told, the WHO when they visited, that samples are not kept for longer than 3 months. So by his reckoning, there was very poor serology in the weeks and months leading up.
Yeah, look, cases existed. It's just not to say that cases didn't exist, but that there wasn't a widespread epidemic in Wuhan before we started to see the hospitalizations.
Could be.
That's the point. Is that we clearly by saying there are primary cases, the first cases clearly aren't the cases of spillover. So the question is, when did it, who were the first people? We don't know.
Yeah,
but
I guess Dominic's point is that the fever hospital didn't have a broad enough coverage to establish any kind of definitive surveillance on a population scale, and also retrospectively that the serology wasn't able to be done because on a large scale, because the samples were destroyed for 3 months. But I wanted to, Jonathan had a hand up, and I also would love to put Sasha on the spot and ask him to ask a question, because Sasha is an expert on a lot of related matters. But the other Jonathan Basile.
Well, I've already asked a question. Would Sasha like to go first? Do you want to ask Sasha first?
Sasha, do you want to ask a question? I would love for you to ask a question.
Okay, thanks. Yeah, I have questions, of course, but maybe Jonathan will ask because he raised arm, and also, I would kind of go after him.
Okay, I'll try to make my question brief. I was curious about what the research you're doing might have to say about political debates around gain of function research. And I'm curious about, I guess I've seen a lot of debates recently around serial passage research, where people are taking viruses that can infect human cells and passing them repeatedly through mammalian cells until they can, or are very close to the point where they can infect human cells. And I guess the argument in defense of this research that's often given is that it'll help us to identify outbreaks before they happen, and can even help us to develop vaccines that could defend against them before they become human pandemics. And I guess I'm just curious if And then, of course, the argument against it is that it could lead to an outbreak. And so I'm curious if this research you're doing, if you feel like it has something to say about that political debate.
And I might, just in the interest of time, maybe ask Sasha to ask at least one of the questions, just because we have limited time, just to maybe get a couple questions on the table, and then Jonathan and Joel can decide which to respond to.
Well, I can answer Jonathan's question really quick. It has nothing to say about gain of function, because it is about the origin of a zoonotic pathogen and not the result of gain of function research. And I can tell you that I'm not a virologist, and the people I work with have myriad opinions on this matter. But the one thing we're united in is that this is not about gain of function. That's a separate debate on a separate topic, and we shouldn't conflate the two of them.
All right, Sasha, the floor is yours.
Okay, thanks. So I have a simple question. So it's you're talking about most common recent ancestor of lineage A and B, and is it correct that your understanding that the virus, this ancestor virus, infected non-human, right?
That's the presumption. It got from bats. It was in bats at some point, and then it was in us, and presumably, it probably went through an intermediate host. That's our best guess. But right now, that is still very much speculation based on the geography and timing.
Right. Yes.
We're not necessarily saying that that lineage B and lineage A were the exact genomes that were in intermediate hosts or in bats, but that the predecessors of them were, and that the most recent common ancestor of a given virus doesn't necessarily have to match the virus that was actually introduced into humans.
Right. Yeah. So when you're talking about that there were two introductions, right? So independent. So you're talking about introductions in human population or out of the original source?
Into humans. I mean, I think from bats into the intermediate host, I don't have the expertise to say how many there would have been. Maybe Joel can comment on that.
Yeah, so we don't, I don't think we have a really good idea about how this virus is getting from bats to whatever host brought it to humans. I think the closer we get to the market, the closer it looks like there was an intermediate host, because there were no bats at the market, and these bats, it's a lot easier to bring sort of a terrestrial mammal all that distance than it is bats, and that just seems to match what we've seen with SARS-1. So I would say we really are, we need more information about sort of bat coronavirus diversity in South China and in Southeast Asia, and more data is coming out, but it's a slow trickle.
Right. Yeah. So, but for this very reason, I did not say that host was bad. So simply just I said it was non-human, right? So in human, it was twice introduced, right? So this is what you basically claim in your paper.
Mm-hmm.
So
yeah.
it means this common ancestor, most recent common ancestor, circulated non-in-human, right?
Of
so
A and B, yes, yeah.
Yes.
Although that's a statement about sort of the ancestry and not necessarily the mutations themselves, because you can have a virus circulate and not mutate, or a virus circulate and mutate for weeks. So, but yes, we believe that the ancestor, or our paper suggests that the ancestor of A and B was in a non-human host.
Right. Yeah. So that is the most important, probably, conclusion of the paper, right? Which is maybe non-trivial, right? So given that the diversity of SARS-CoV-2 relatively small compared with the distance to the closest non-human host or virus isolated, right? So distance is much bigger, right? So
Yes, yes, it is. It's what about 400 mutations back to our sort of hypothetical bad ancestor in aggregate across the whole genome, although
Right.
um,
Right. Yeah. So like you said, and Jonathan said that it is actually based everything on the two mutations, which is shared with the lineage, if I correctly remember, A and B. Yeah. So which is against 400 differences, but still two, because genome size 30,000, it's considered quite improbable that they emerged independently, right? And so because of this.
yes, it's that and that if A were the ancestor and we didn't know about the bat viruses, the molecular clock
Right. Yeah.
wouldn't put it there. So it's also this sort of this broken clock in the beginning of the pandemic that I think has gone a little underappreciated
Right.
before now.
Right. So it's based on the models you used. It is very unlikely because of this type of conclusions. But of course, the question is whether its models are correct applying there. So this is a big question, of course, right? So because we still have to acknowledge that viruses can do many things and pretty unusual things. And of course, convergence could happen, although, I mean, it's how we could talk about this lineage A and bats, and so this is very different. Right?
Oh yes. Yeah, and we think we're seeing convergence to bat-like viruses across the tree, even today, even within the different variants of concern, we're seeing an exceptional amount of convergence to bat viruses. Now, one thing I will say is that separate from all the models, if we just take a step back and we look at coronaviruses in general,
Right.
SARS jumps into humans multiple times, 2, 3, probably more. MERS, I mean, it just there is no human tree. It's all just camels into bats. This new Delta porcine coronavirus that they found in Haiti, multiple jumps into humans. You look at the civet farmer, or sorry, civet sellers in markets around the time of SARS that never had SARS illness, and 73% of them have SARS-like and serological reactions. There's, we're clearly being pelted with these viruses. So the idea of like, well, how much more, just from a sort of naive perspective, is one jump or two jumps or three jumps or four jumps, what's to be expected? And I guess my perspective is that if you put a virus that's capable of transmitting among humans, human adjacent, you're going to need a lot of jumps to get it into people, first of all. And then once it gets in once, there's really not that much to stop it from getting in again, besides another human and another exposure to a reservoir we already know is in close proximity to humans. So I think from a naive perspective, it may be more unlikely to only see a single exposure lead to a pandemic of a coronavirus than multiple exposures, one, two, or more of which eventually take hold in humans.
Right. Yeah. So, but Joe, here actually two different questions. It's one question about how many times zoonotic transmissions in this virus species, and what happened actually at the beginning of SARS-CoV-2 pandemic. Although they related, but it is different. So I completely agree with you. There were many transmissions, and most of them were dead end. And probably in 21st century, in 20th century, and 19th century, so we simply don't know. So what we have a record, actually two pandemics for SARS-CoV and SARS-CoV-2. And so that is, but it does not mean that there were not many. So, but it is a bit different question when you claim exactly that at the beginning of SARS-CoV-2, there were two transmissions, although they in agreement with the general picture and assumptions, because what we see, it is mostly the dead end, even during current pandemic. This is absolutely, I mean, it's no hypothesis. It's simply just mostly trivial, although maybe not broadly acknowledged, but it is almost trivial. But it is, of course, it is very important that there were two transmissions, like you claim, which established this pandemic, although one lineage played probably dominant role compared with another one. It's possible, I mean, from theoretical point of view, but of course, questions about evidence, right? You presented evidence, but how it is solid and whether it can be independently verified, which is very difficult, probably, right?
Absolutely. And I would say one of the predictions of this sort of multi-zoonosis hypothesis was that lineage A was actually present at the market at low levels, something
Right.
that when we conducted this study and wrote it up, there had not been shown. And then I think 14 hours before we published our preprint, the Chinese CDC confirmed the prediction that lineage A was in fact at the market at extremely low levels. So you're right. It's a hypothesis. It makes a prediction, and that first prediction was borne out. So I would say it is the as I've described it to people, it's the most complete. I would say multiple jumps into humans is the most complete and comprehensive description of the early genomic and geographic diversity. Did it necessarily happen? No. Are we waiting to see if they can confirm with more sequences and samples? Yes, but I have yet to see an explanation for the early genomic diversity that explains all of these quirks with the clock, with sort of the giant explosions of diversity at the base of A and B, with the differences between A and B, and the sort of the presence of them both at the market, and the fact that A is infecting people closer to the market than B at the beginning of January. Now, if there's an alternative hypothesis that can explain all of that as well as two independent jumps, I'm all ears. But for now, I think this is our most comprehensive explanation of how the pandemic began.
So may I ask you one question, Evan?
Please.
So is it possible to apply a methodology also to learn about origin of Omicron?
I think that the question there, I think, is a little bit different with Omicron. I think people are wondering about what the origins of Omicron were, and I think we definitely need more data to figure that out. I think we are trying to work on a few other methodological techniques for that question, because the ones from our paper are helpful, but are not sufficient, I think, to answer that question yet.
Right. Yeah. So I asked because, as you all, of course, know, of course, Omicron is different, right? So compared with everything else, and it happened just before our eyes and actually escaped our attention, right? So it definitely happened during a short time period, relatively short, and so in a way you could compare time frame. What happened at the beginning of SARS-CoV-2, probably lineage A and B, or to some extent probably, what happened to Omicron, right? And so you could see from this example already how it's possible to generate something that absolutely looks like probably not easy to fit in the conventional model. So you need something extra, right?
Yeah. So I was just to jump on what Jonathan was saying, with Omicron, you have a lot of adaptation very quickly, and clearly sort of the phenotypic result of that adaptation, you can get reinfection with one, Omicron subvariant after the other. And then you also have sort of this signal of recombination between them, which implies something else very interesting was going on. Now, whether that's sort of within long-term chronic infection or within sort of a jump back into animals and then spillover of Omicron. A lot of speculation out there right now. I think something similar, but one of the aspects we're able to how we're able to estimate the time prior to these spillover events is relying on their not needing to invoke adaptation, or knowing that if there was adaptation, it actually doesn't impact the timing very much. But with Omicron, because there's selection and sort of strong phenotypic divergence, it implies, I would say, something much more biologically interesting leading up to Omicron than what is essentially neutrality at the beginning of the pandemic.
Yeah. So, but that is
but your thinking is you're onto something. I think there's a path here. I just don't know what it is.
Mhm. Right. Yeah. So, but again, so it is assumptions in a way. So, of course, we know much better conditions when Omicron emerged. So, because we already were in the pandemic, and so this adaptation, it is kind of basically it is background. So you could, but we actually don't know what was background in the during the spillover. So, and that is sampling, of course, much smaller. And so, and of course, looks like humans, or I mean, this is transition. It is so unique event. We don't have statistics. We don't have much knowledge to refer to about transitions, right? So it makes much more difficult to study. Is it correct?
Yeah, I mean, I think, right, like with SARS-CoV-2 origins, right, there's a bit of a dark spot, and we're, like Joel said, we're operating under neutrality. But I think even though that we have a lot of sequence data and a pretty good understanding of what the diversity of SARS-CoV-2 is across the world, clearly with Omicron, there was still some sort of dark spot that we don't understand, whether it is extreme adaptation in one or several humans, or whether if it was a reverse and subsequent zoonosis event. Like, that's also, both cases are still dark spots that we don't have great understandings of, even if the stuff outside of that we do have an understanding of.
Right. Yeah. Okay. No, it's of course I could ask more, but of course I don't. Yeah. So
We
thanks a
should
lot.
let you
Yeah.
start, Sasha. We should let you
Right.
you know yeah, but I feel like this could go on for much longer. I'm happy to stay a little bit longer. Joel and Jonathan, what's your next 15-20 minutes look like?
I can do another like 15 minutes.
Okay.
I'm here.
All right, Sasha, keep going. Oh, you're muted, I think.
Yeah, but okay, so but of course, important questions. It already was raised. It is about role of market, and so that is also it is a central event if it accepted, and there is evidence, of course, a role of market. But if it's not market, and even it's not Wuhan, so then it makes much more uncertain probably picture, right? So because we're talking about hospital in Wuhan, but if it happened first cases outside Wuhan, so we have to consider a much bigger area, and also sera of the people have not been kind of studied there, so it's completely different ball game probably.
Yeah, although I will, yes, although I will say, I think the most helpful result from our two investigations into this that Jonathan alluded to earlier was the idea of how many people can be infected before you have to have the genetic ancestor. And I think our best estimate now is if a few dozen people can get this virus before by what the earliest estimate would be in mid-November. So you can't have hundreds of people elsewhere in the world get infected and not but before that date. Now, you're right, you wouldn't see very many people now, and so based on the sort of molecular evidence alone, you could bring it into Wuhan. And I know there have been discussions about a case on November 17th in broader Ube who was hospitalized. Of course, that article also went on to claim that there was a case every single day diagnosed through December. And then, of course, the WHO didn't find evidence for the veracity of those cases being COVID rather than, it just being a particularly bad flu season or other cases of pneumonia. So it's very difficult to have like I said, you can't have it circulating widely elsewhere. And then I think the coincidence of it just ending up at an animal market is this is not one of the most highly trafficked areas in all of Wuhan. It is just, it's just a wet market. It's not the only wet market, and it's not a major sporting event. It's not a shopping mall. It's not a university. It's a market where the earliest cases all trace back to the part of the market where they sold mammals we know to be susceptible to SARS-CoV-2 infection and capable of transmission. So I think yes, is it possible before the when the WHO report came out and they said, well, there were no animals sold at the market, I was sort of that mindset of like, well, maybe, someone got infected in rural China and sort of brought it in, directly from a bat and brought it in. But then, the animals were there. The animals were there in November, and then the animals were gone on January 1st. And that just, I mean, there's just a very natural progression of events here. It doesn't require sort of broad conspiracy. It doesn't require, it just is the same thing we've seen, basically the same thing we saw with SARS happening again. And I think anyone who was there on the ground at the time realized exactly what was happening.
All right. Yeah, but Joe, yeah, it is highly plausible. And I mean, no question, it's right. So, but also, what I recall, my impressions 2 years ago or 2.5 years ago, of course, sensitivity to emergence of wars, which will be similar to SARS-CoV, in China was very high, unlike in outside the world. So, because there was imprint in the society already. And so there was imprint also to action. So what you mentioned, actually, the change in the market, it was natural, you could explain this by natural response of the authorities. Regardless what happens there, it's simply just because they knew that it already once happened, and so then we have to kind of remove. But whether it happens there, it is a question. So another aspect, of course, when we're talking about, like you said, that how many people had to be infected before we kind of detected. So important also characteristic, of course, as you know, it is population structure. So if you just could imagine that first we infected very young people, it could go for many 10,000s or 100,000s. I mean, just an example. Probably, I don't know. So it depends. So it's, of course, changes will happen, but at least there will be no sickness. So
there'll be no sickness, but there will be a genetic ancestor.
Changes, but could be neutral changes, right? So of course it's restricted in terms of, but it will modulate actually, and also will allow you to involve bigger area geographic, right? So it very much depends who you're infected. So
that is true. Although some of the earliest hospitalized cases were, they're not elderly. They're what are they, in their 40s and their 30s. I mean, these are not old people who got, who were the first hospitalized cases, especially those linked to the market. Also, if you have 10,000 young people infected, we would see the evidence of that in the genetic ancestor.
Mm-hmm.
So clearly, even if that were the case, that would have happened in November, and then it would have had to have spread from there. Now, alternatively, if you have this sort of spillover in a rural area where, which it could happen, and actually as Jonathan's previous paper showed, the most common result of a spillover in, say, rural China is extinction. And it's not just extinction, 70% of the time, it's extinction 95% of the time, 98% of the time. This is like, I think the analogy here is like, why did we see Ebola twice in 1976, as if it just decided to start emerging in humans, rather than we just started noticing it and it has been, there since time immemorial. So yes, it is possible that the first people got infected. You just can't. It can't be thousands. It can't be tens of thousands. Um, before the genetic ancestor, and that doesn't matter what age group it's infecting.
Actually, on that point, it can't be thousands. Like, so part of what, historically, kind of sociologically, clinically, we're trying to think about is just the conditions of recognizing, noticing, and sequencing these early samples. And, part of what we're learning as oral historians, as, people who are really trying to attend to the dynamics of power within a hospital in China that's, recognizing this emergent disease. You don't want it happening on your watch, and so that's part of it. And also, again, that point I made earlier, just the very limited capacity of the, and again, it was an exceptional, much better than the facilities that we have in the States or Europe for recognizing, identifying, and kind of doing something about an emergent infectious disease, but still a tiny subset of the population is getting shunted into these hospitals with
Sure, but the genetic ancestor is a measurement separate from the hospitals. Now you can think, so an analogy here is the HIV pandemic, which we didn't start noticing until 1980, 1981, but clearly started spreading widely in Kinshasa around the turn of the 20th century. And the genetic ancestor traces back to that time of explosive growth because it's captured. It doesn't, and now also important to know that there was some time in between the zoonotic event in southeast Cameroon, then it gets to Kinshasa, and then once it starts to explode, we have these lineages of HIV that persist to this day. And we can trace it back to say this is where it happened, and that yes, there could have been low-level transmission before that, but there wasn't widespread along the rail system in what was then Zaire. All of that can be found in the genetic reconstruction. And granted, that's a century old instead of in their cases 2 years old, but the principle remains is that once you start having sort of the spread of virus, that is captured in the genetics, and if you go back to that point in time, essentially it doesn't disappear. We have those lineages still circulating in humans today. There is still some very, very small amount of all of those descendants. I mean, granted, almost all of it's Omicron, but not all of it. So we could have missed it in terms of our infectious disease surveillance, in terms of our serology, in terms of hospitalizations. But what Jonathan's simulations show is that there is only a very, very small amount of time, days, weeks, maybe a month, that we could have missed it in the genetics. And then after that, if this virus were to go pandemic, it would have to have established that genetic signature. Otherwise, it would go extinct. And that's sort of where we say, there's no room for cryptic spread because the virus is stuck between a rock and a hard place. Either it leaves the genetic evidence behind or it goes extinct. And since it took off, since it diversified so quickly, and you get sort of those massive bursts of transmission early on, which we see in both A and B, all of that says there's just no time for widespread cryptic transmission. You could have low level. I mean, according to the genetics, of course, it's not consistent with what we know about the epidemiology and the first hospitalizations in the market. But even if you throw all of that aside, you could have a single person infected just based on the phylogenetic tree in October. And then very, very low stuttering transmission until November. but you can't have widespread transmission in October, early November, because you don't see it in the phylogenetic tree. So you get one or the other.
I would also add to kind of bring in the other preprint, Michael Warby's preprint, where they look at the geographic radiation out of Wuhan, elsewhere. The common analogy there now is like a drop in a pond, and it slowly radiates outward. We see that the drops of lineage B and A are at slightly different times, so that's just more evidence toward multiple introductions. But I think we can actually take that analogy to genomic diversity as well. The genomic diversity radiates out of Wuhan. It doesn't radiate out of somewhere else. And if there was really widespread, our genomic diversity wouldn't basically also be radiating out of Wuhan, if not the Huanan market itself, right? And so it's hard to imagine hundreds, if not thousands of people infected, not leaving such a trace of genomic diversity elsewhere, where the concentric circles radiating out of the Huanan market wouldn't exist with such fine resolution if we had more genetic diversity in places outside of Wuhan.
For me, it's kind of still a question of sampling, but Sasha, do you have any?
I'll leave the sampling to Mike Warby and all of them. I'll talk about the genomics.
Yeah, but just a very limited set of samples from one particular city. Sasha, do you have any final thoughts or comments?
No, I mean, of course, what you presented, it is probably the best kind of what can be done kind of with this type of data, right? So that is a question. So, but still, of course, questions could be asked. For instance, if this transmission first time zoonotic happened outside Wuhan, where density population was smaller, of course you could explain that Wuhan, of course, density, and so then what you could see because sampling is there. And I mean, this is probably impossible to discard. Yeah, so it's and time frame. I agree completely. Genetic, of course, signature is there. So of course we're talking about a small time frame, right?
So yeah, first of all, your first point there is spot on. If this virus, because presumably animals, they got to the market somehow, and they probably got infected, before in the market, presumably in the farms, But so let's say hypothetically you have an infected group of animals on a farm in Enshi, this sort of more rural prefecture. What happens when the virus jumps into humans there? Well, the most likely scenario is that the person who gets it gets a mild cold and doesn't infect anyone else. And that's the end of it. And a dead end transmission, similar to all of those palm sellers in 2003, they never even knew they were sick. So I think you have repeated spillovers, and it's only by having sort of some takeoff in this metropolitan area that leads to a pandemic. And that if we're constantly being pelted with zoonotic viruses in rural areas, that's not going to lead to a pandemic. So we just don't see them happening. And if you have a related virus that isn't quite as fit, because keep in mind, SARS-CoV-2, it's fit enough to cause a pandemic, and yet still its tendency is towards extinction. So if you have a related virus that is slightly less transmissible, it goes extinct even more frequently. And actually, in our previous paper, although we sort of put most of it in the supplement, we actually modeled what happens if SARS-CoV-2 has to evolve into a more fit variant before it takes off. And the problem is that a less fit SARS-CoV-2 goes extinct really, really quickly, and and the less fit it is, the shorter amount of time it has to adapt and evolve. And the more fit it is, the more it just looks like regular SARS-CoV-2. So you don't even have this period for cryptic transmission of a less fit variant because we know it went extinct. So either it adapted or it died out. Now that we are seeing more evidence for two spillovers, it actually seems like SARS-CoV-2 didn't need to adapt into humans in the beginning. It was good enough because if A and B both spread separately, and they're both two separate jumps, they're both you don't need a period of adaptation within humans. Now, that said, D614G shows up pretty quickly, and then we're off to the races with all the variants. And then, of course, A has picked up its own D614G multiple times. So, it's not like SARS-CoV-2 isn't evolving and adapting and getting better at spread. It just, evolution doesn't need best. Evolution just needs good enough. And, based on what we see now, SARS-CoV-2, when it first got into humans, appeared to be just good enough. And now, unfortunately, is much better.
I'll also add just one final point, and then I actually have to jump off.
Yeah.
With respect to sampling, where you think that we're only seeing kind of the product of sampling, if we were to assume that there were wide cryptic spread, again, elsewhere, right? Bringing it back to both Joel's point where we leave markers and my point where there's this concentric spread outward from Wuhan, we should not only be detecting the descendants of Wuhan or descendant lineages from Wuhan elsewhere in the world. If there were long-term cryptic spread in Italy or anywhere else, that should then show up in the data because that would still be there later. It wouldn't have disappeared and then we only see stuff from Wuhan coming in later and then we only sample that. That's why the sampling elsewhere in the world isn't consistent with it because it still looks like it just came from Wuhan.
Sasha, do you have any final thoughts? I know that you got to go. Maybe I'll just make a broad point is, I think we're reaching similar, I guess interested in kind of different sites. And I guess the site that I'm interested in, or the kinds of sites are the places where these viruses are circulating and have been circulating for decades. And, I think it's kind of just different emphasis and different, like in thinking about the emergent event, like, to me, what matters is like, how, this virus learned how to get around in people somewhere, and it probably wasn't the Wuhan market. So that's kind of one of my working assumptions. But Sasha, do you have any final thoughts?
I enjoyed very much and learn from you, from Joel and Jonathan. So thanks so much. But also kind of following what you told, Joel. So if you look for MERS coronavirus, where we could see multiple transmissions to humans, right? So at dead end. So basically, you already have a proof that how it could happen with anything else, which we kind of don't know if infected in good shape. And so, for instance, right, so young people, or farmers rarely kind of infected. So it could go undetected in terms of hospital, and nobody basically took genetics there. So that is one, of course. And of course, question it's about how many people were infected, and whether if it a lot of people, we should have kind of seen additional lineages. It depends, of course, our relationship about extinction and transmission, right? So again, if it happens in rural area, when transmission could be easily broken, so because a chain of transmission. So in theory, it could happen kind of for some time, and this would be then unless it got in the metropolitan Wuhan, right? So then you could see everything. So and you already mentioned about Great Britain, so and other areas where dead end happened, even kind of in our world, which is well connected, right? Yeah.
So thank you both for sharing this paper. It's important research. Thank you for letting us ask the tough questions. I hope you come back. It's super important, and I think we need to keep having these conversations to push the research agenda forward. So thank you so much for coming. Again, please come back, and hope to see you in a Zoom room or in real life sometime soon.
Yeah,
thank you for having
so
me.
much.
Thank you.
And Frederic, I'm gonna send you an email with my number. So let's meet up in Milan. Oh, you're muted, I think.
Yeah, right. Yeah.
Where are you at the moment?
I don't even know, but I'll text you my number. And Sasha, I would love to have a proper catch up sometime soon, one-on-one.
Okay, I'll call you by WhatsApp.
Okay, ciao, ciao.
Ciao.
Bye.